Hello friends, welcome to my YouTube channel New Biologist. I'm going to discuss in brief about the rDNA technology. So it is also been considered as a called as like genetic engineering, genetic manipulations or in a gene cloning. So what is we are looking to do here? That is like a simple, you can say that like additions, deletions or manipulation of a single trait in an organism to create a desired change. And how it is been done so you can say it's like a copy and paste DNA from one individual to another organism or another individual. So it's a simple copy and paste mechanism. So what is the purpose? Here we are looking to transfer the material or genetic material from one to another individual for a specific desired things. We may take the one of the example like the human genes, maybe that was been introduced to the bacteria. One an example is like insulin. Then it can be possible that human gene can be inserted into animals. Also, like a bacterial genes can be inserted through plants. So any place, horizontal, vertical, any of you can say the transfer of the genetic material can be possible. So what are the stages are involved? So there are the five stages are there: isolation, cutting, ligation, and insertion, transformation, and expression. Let me introduce about the isolations. That is from the two places. That is from the host and like a isolation of specific gene from the donor and the host. So one is like a let's take one of the example that is a human is a donor. So first need to isolate the DNA. We may need to design the probe or use the primer to get the desired fragment, or we may go for the restriction enzyme even to get the desired fragments of our interest. Another is a part is like a bacterial cell from where we are looking to do a certain like a cloning. So intrachromosomal DNA is generally isolated. And second step is like a usage of restriction enzyme. What is there? There you can see the molecular cutter. So they are going to cut the DNA very precisely from the end to generate some sticky end. So we are going to cut the donor DNA as well as we are going to cut our plasmid. So what we are looking to do, that is like a sticky end are now possible on the donor DNA as well as sticky ends are there on plasmids. So let them mingle with one to another. So we are going to use the molecular glue, or it is called the DNA ligase. So it is going to create the bonding, the covalent bond going to form by you can say the DNA ligase. So ligation and insertion, you can say like a foreign DNA is being cut with a desired fragment. Right that time, plasmid it is being cut and are joined by the DNA ligase. So the finally the plasmid we are going to get is called hybrid plasmid. And this hybrid plasmid are transformed to our bacteria where we are looking to get or synthesize our product. So at the end, we are looking to study their expressions. That bacterial cell who has received this uh, DNA, our the desired fragment in a clone part, should able to do reproduction. And after the reproductions, they should able to produce our desired protein or the product. The summary of the steps, you can say the cutting with the restriction enzyme, that is a donor DNA as well as like a plasmid DNA to ligate them and create our recombinant DNA that we need to transform and to get our desired product. Some applications, let's see, is like production of humulin. It's generally been used in the diabetics. We very well known about it. Another is in plant applications. If you are looking, that is a golden rice. That is like you can say the possible solution to vitamin A deficiency and even into vaccines. If we are able to engineer some of the uh, antigenic protein to the any bacteria. So they are going to synthesize continuously some antigenic determinant, and that can be used as to vaccinate the human being. One of the success story you can say, or the live example, it is the vaccine against the hepatitis B being synthesized in this way. Thank you.